Hello, my name is Onwe Reuben Ogba. I'm a graduate of the University of Nigeria, Nsukka, where I obtained my Bachelor of Science degree in microbiology. I'm currently a research assistant in the Department of Microbiology, University of Nigeria, Nsukka. I'm here on behalf of my co-authors to present on the topic phage-derived proteins against common nosocomial bacterial infections at the 4th Asian Conference on Science, Technology, and Medicine. Bacteriophages, also known as phages, coined by Thierry in 1917, are those viruses that infect and kill bacteria. And before the discovery of antibiotics penicillin in 1929 by Alexander Fleming, bacteriophages have been in use for therapeutic purposes, especially in Europe. Recently, a great threat has been established globally due to the emergence of multi-drug-resistant bacterial infections. And this is as a result of a variety of factors, such as wrong use of prescription and misuse of antibiotics, indiscriminate spread and agricultural use of antibiotics, and as well as positive selection of multi-drug-resistant genes gained from prokaryotic exchange of genetic materials. In the United States alone, the Center for Disease Control and Prevention estimated that about 23,000 persons die yearly due to multi-drug-resistant bacterial infections, and this may double in developing countries. As a result, scientists have recently keyed in innovative research on the use of lytic phages and their proteins as antimicrobial agents, and they are promising alternative to conventional mothballed antibiotics. This study aims to summarize using available literatures on the phage-derived proteins used to combat common nosocomial bacterial infections. And for us to understand the backdrop, the science behind the use of phage proteins, we need to understand the interaction between the phages and the bacterial host. The bacteriophages, as earlier stated, produce an array of proteins to successfully infect the bacterial hosts. And they play specific roles from host recognition through takeover of host cell machinery to cell lysis for new virion release. And based on stages in life cycle, phage proteins can be classified as either morphogenesis proteins, which are used during the phage development, the genome replication / regulation proteins, and then genome packaging proteins, host cell lysis protein, and all hypothetical proteins. However, the last two classes are of most therapeutic importance. Let's look at the image. The image here, let me make a zoom. Okay, this shows the phages and their cell wall bacteria target sites. If you look closely, you see that there are majorly two classes: the depolymerases or phage-associated polysaccharide degrading enzymes, that is PAPDE, and the lysis cassette proteins. Both are collective called the protein receptor binding, phage receptor binding proteins. And the depolymerases, the phage polysaccharide depolymerases are often associated with tail structure of the virions, such as the tail fibers, the tail spike, base plates, or the tubular proteins, and sometimes can be located at the neck or the collar region of the phages. And phage apply different depolymerases to enzymatically degrade the capsular polysaccharides, example, the arginate and polycylic acid, and or structural polysaccharides, such as the lipopolysaccharides and their peptidoglycans. And depending on the side chain degradation, phages depolymerases are divided into 5 majorly. These polysaccharides here, depending on the side chains where these depolymerases attack, we can find about 5 types of phage depolymerases, which include the peptidoglycan hydrolases, the endosalicidases, endoharmonicidases, the arginate lyases, and the hyaluronate lyase. And these polysaccharides, so this is the polymerases, specifically cleave the glycosidic bonds in bacterial extracellular polysaccharides. These extracellular polysaccharides, you can see here, the capsular polysaccharides, the free extra polysaccharides, the lipopolysaccharide, and the extra polysaccharides. And thus differ based on type of monomers linked by these glycosidic bonds. Okay, we have the lysis protein, or the lysis cassette proteins here, and we have three major, the holins, which act on the inner cell membrane, have the lysine that act on the thin peptidoglycan layer, mostly for gram-negative bacteria, and the spannin, which act on the outer membranes. Okay, it's a lot of studies so far has been done to describe these phage lysines against common nosocomial bacterial infection. However, few holins, such as GH15, have been isolated and been described. And so far, holins, no holin has been described to actually have been described so far. And the term, sometimes the most described lysis cassette protein is the lysine. And the term lysine and endolysine are often used interchangeably. However, those that act on the peptidoglycan after phage absorption to the host cell surfaces are known as the virion-associated lysine, or the virion-associated peptidoglycan hydrolases, or the tail-associated lysines. While those that act during the progeny release are also called the lysine, and also that is called holin lysine spanning system. Okay, having said this, let me minimize this. Okay. The research shown so far that for phage proteins to be used, phage proteins, both natural and engineered proteins, to be considered as most excellent antimicrobial candidates against common nosocomial bacteria pathogens and other bacteria, it must possess at least two in any of the following ideal functional properties. And they include, it must have independent catalytic domain, must have or has putative antimicrobial peptides. It must have positively charged amino acid on either the catalytic domain or the putative domain that confers cell membrane destabilizing properties to either the CD or the PD domains. And this is to lyse, this is lysines. Okay, the third properties, which is against positively charged amino, it must have positive charge, and are mostly found in the gram-negative bacteria, those phage proteins that act on gram-negative bacteria. And lastly, it should possess host cell receptor binding proteins. And these are most common or commonly found in those bacterial phage proteins that are used against gram-positive bacteria. Okay, as you can see, this is a table example that summarizes the phage proteins that have been discovered and described. However, I presented one example here, like lysine EFM5 on the protein on the parent phage is derived from IME A5M, and you can find the accession number KT5888072. The structural characteristics is the enzyme is modular with amidase activity in its N-terminal domain, with putative catalytic residues, E19T138, and putative zinc binding residues, H27H32, and C40. Okay, there's no biological characteristics has been described, and there's no modification for this. Research has not been done to that towards that. And activity, this phage protein has been found to be very effective against Enterococcus faecium and vancomycin resistant Enterococcus faecium. The dose found was 50 microgram per ml. And in vitro findings, it is active against about 82.65% of Enterococcus faecium strain tested and 7 vancomycin resistant Enterococcus faecium strains with minimum inhibitory concentration range of 16 to 120 microgram per ml. Okay. Thus, we have the major challenge so far that has been found in the use of phage proteins against nosocomial infections are the phages and phage proteins are still classified as high-risk medication due to possible endotoxin release into the bloodstream after bacteriolysis. And because of the high specificity, it is difficult to identify the exact phage protein or proteins needed to treat a particular bacterial infection. And this is one of the major challenge faced that the scientists are really facing in the discovery of phage proteins. And a lot of studies are needed to establish the pharmaceutical, so the pharmacokinetics and the pharmacodynamics of the phage proteins. In conclusion, phage proteins may in future prove to be a valuable component in a multi-pronged approaches to dealing with the worsening challenge of increasing antimicrobial resistance in bacterial pathogens. However, more evidence from high-quality clinical trials will be needed to increase confidence in the breadth of the effectiveness and regulatory framework for licensing these biological therapeutic agents. Thank you for your listenership.